All right. Hello, everyone. My name is Ryan Stumbaugh. I'm a PhD student in the Northup's lab here at UNM's biology department. I'm very excited to be contributing my own talk today in this great lineup of presentations, especially the one we just had. But this is my first talk at an NSS event and my second talk on these data. So go easy on me. Okay, be patient. My heart's pounding, hands shaking, all that good stuff. All right. Let's get into it. A microbial symphony of nitrogen cycling within lava cave environments. I had a lot of great help from Joseph Medley, Jennifer Hathaway, Diana Northup, and my co-advisor, Cristina Takacs-Vesbach, who contributed a lot of information and advice when we were thinking and planning about this project. So why would a microbiologist investigate lava caves? Well, they're an invaluable reservoir of novel microbial life forms. So most or significant amount of these sequences in DNA that we get out of our samples are undiscovered. And that's pretty exciting, right? This gives us great information about new environmental roles that these microbes could be playing in these environments and sort of add to that tree of life that Jenny was talking about in her presentation, filling these little gaps of microbial roles when it comes to environmental microbiology. Another great point is the source of novel antibiotics and enzymes. So 70% of our antibiotics come from a group of bacteria called Streptomyces, and we have a ton of Streptomyces bacteria in these cave environments. So by understanding, how these microbial communities establish themselves and flourish in these systems, gives us a better idea of how to analyze these other valuable groups of bacteria that are also growing in these communities as well. These guys also contribute to secondary speleothem formation. So these bacterial and archaeal cells, they'll produce byproducts such as bicarbonate, and their overall negatively charged membranes will attract positively charged minerals such as calcium. When you have calcium and bicarbonate, you mix those together, you get calcium carbonate, and they precipitate these minerals on their membranes, and they add to these different speleothem formations, making these very beautiful structures. And it also tells us ways about how life may be able to flourish or persist in other rocky planets such as Mars. So when we think about Mars, it's pretty hazardous conditions. You've got cosmic radiation, pretty intense weather patterns. So a good place for life to hang out would be inside of a cave. And so studying how these microbes can live in these lava cave environments gives us an idea of how they may be able to do so on other planets such as Mars. However, basic knowledge within lava cave environments regarding nitrogen acquisition and cycling is limited. And so we wanted to explore this a little further and see how these microbes are achieving these processes. So the importance of nitrogen. When we think about what makes up life, including us and basically every other life form on this planet, it's a lot of macromolecules, some of them being DNA and proteins. And when you look into the structure of DNA, you've got your nucleic acids attached to a sugar phosphate backbone. And then in proteins, you've got series of amino acids. And I'm not going to really drill you guys with too much biochemistry. The main thing in these figures is all of the nitrogen atoms in these structures, right? We've got some here in the amino acids. We've got some in the nucleic acids of DNA. So without nitrogen, you don't have DNA and you don't have proteins. Without these two types of molecules, you don't have life, okay? So that's why it's so important that we're looking into how they're sourcing their nitrogen. Without these mechanisms, they can't establish themselves or flourish in these environments. And taking it a step further, we're going to go into the nitrogen cycle. So this is a loose nitrogen cycle, but it normally starts with N2 gas that is then fixed into ammonia. So you've got our blue nitrogen fixation here. Then ammonia is oxidized into nitrite. So we've got our ammonia oxidation here. Nitrite is further oxidized to nitrate. So we've got our nitrite oxidation, and then nitrate can be reduced back down to N2 gas and other nitrogen compounds. And then this cycle can continue. And the reason why this cycle is so important is because some microbes need different forms of nitrogen compounds. And so this cycle can transform them and make them available for bacterial or archaeal species that need ammonia versus others that need nitrite or nitrate. So it's really important to sort of study how this cycle is conducted by these bacterial and archaeal species in these environments. And we're really curious as to how they may be working together to achieve this process. We know that bacteria can work together, but interactions between bacterial and archaeal species in this context, in these environments, is fairly limited. So we wanted to dive into it a little bit. So here are the research questions that me and my lab group sort of considered. So where are bacteria and archaea sourcing nitrogen, and how does their usage of these sources fluctuate? Do archaea and bacteria engage in cooperative interactions to achieve nitrogen cycling goals? And which secondary mineral and microbial deposits are nitrogen cycling hotspots? And before we go any further, I guess just to reiterate what Jenny was talking about, archaea are sort of the ancient cousins of bacteria. They're both single-celled organisms. They're not eukaryotes. And so we really were curious about how they could be working together because we have a good amount of bacterial data, but archaeal data is fairly limited. So we wanted to really explore that. So our study system is the Kipuka Kanohina Cave Reserve on the Big Island of Hawaii. And it's sort of this area on the southwestern region of the Big Island. These caves are very beautiful. It was a real privilege to be able to go run around in them and check out all the different formations and conduct our studies here. The abiotic conditions, we've got 66 to 69 degrees Fahrenheit, but don't let that mislead you. It seems fairly cool, but it is very hot. I was very slimy, very slick with sweat as I was running through these lava caves. And I think at one point I asked Diana, what about the contamination, right? I've got all kinds of sweat coming off me. And she kind of gave me a look in response that said, don't think about it. And we're not going to talk about that anymore because it is a serious issue. These lava caves are composed of thalatic basalt, meaning it's saturated with silica, and it's got iron and calcium, other types of minerals. It's high humidity and minimal rainfall, and it's about 330 to 415 meters of elevation from the ocean. So the residents of these lava caves include insects, wild cats, and dogs, some of which I've been chased out or from caves on the Big Island. We've got some wild dog populations that like to hang out near the entrance.
That's for a guy who feeds his children.
Yeah. So, yeah, not sure if Diana, didn't like me as a grad student, sent me down to these entrances, but yeah, right, great idea. But they also have old remnants of agricultural practices. Farmers used to have goats and other animals sort of hang out in these lava caves as areas to store them. So some lava caves near the entrances, it sort of looks like dirty white carpet from all the goat poop and other animal poop and just waste that they've left there. So you kind of got this nice carpet entrance in some caves as you're walking through. Yep, exactly. Yeah, really good nitrogen. Yeah. Not deep, not quite as deep as we want though. And then we've got plant roots come from the surface. Now there is low amounts of vegetation above these lava caves, but they do have roots that protrude into the lava caves themselves. And these can be potential sources of nitrogen and just nutrient influx into these systems in general. All right, so across the cave preserve, we went into 3 different caves. We took 37 samples in total, and from each cave, we took 12 to 13 samples. Of these 12 to 13 samples, we've got 3 coralloids. You can see up here, we've got 3 microbial mats and 3 mineral crusts, then 3 moonmilk samples. And thank you, Kenneth, for these wonderful images, and Diana as well. We wanted to make sure that we got a wide variety of sample types when we were really trying to figure out, how these microbes are cycling their nitrogen and what these communities may look like and how they may differ across these sample types. So the lab work, we extracted the DNA, we brought it back to UNM. We sequenced the fingerprint 16S rRNA gene, and essentially what that means is kind of exactly what it sounds like. It's the fingerprint gene. So when we look at this gene, it tells us the different species that we have in our sample. It is unique like their own fingerprint. And then the data was analyzed using UNM's supercomputer to run all of our fun analyses and get our fun little plots that I'm going to subject you guys to right now. So don't panic. It's okay. I'm going to... Yeah, Diana tried to help me out with this. We're going to go through how to sort of interpret these plots. These are differential abundance plots. And so we've got our 4 sample type groups up here at the top. We've got coralloids, microbial mats, mineral crusts, and moonmilk across all 3 caves. And if you look here on the right, these are the major genera or major groups, in this case of Archaea, that we saw across all of our sample types. So when you look at one of these vertical columns here, this represents one sample. And then you've got the different major groups of bacteria present in those sample. And some of these colors do repeat. So when you see something like this, where you have this blue up here and then this blue at the bottom, you just kind of look and see the first blue up here is Candidatus nitrocosmicus. We know that this is This blue correlates with this one, and then this blue down here is the unclassified Woes Archaeales. So it kind of goes in the order from top to bottom. So first off, when we look at the major groups that we found of Archaea within these samples, we have a ton of unclassified Archaea. And this is really exciting for a grad student. You get to come and look at these samples, right, and say, wow, some of these have never been seen before. And particularly this one, this unclassified up to the domain level. That's insane. There's still 7 more taxonomic levels below that. That's sort of like finding an animal and not knowing if it's a monkey, a snake, a bird. You just know it's an animal. That's it. So we have no clue what this guy could be doing. But going back to the nitrogen cycling, these gold AOs represent ammonia oxidizers. So we have a ton of archaeal ammonia oxidizers in these samples. And you can see they're pretty prevalent. If you consider Candidatus nitrososphaera, you can see it in every single, or almost every single sample across the 4 sample types. And we do have this one nitrate reducer down here, this unclassified Woes Archaeales. Thanks to Diana, she was able to tell me that Woes Archaeales is pretty associated with nitrate reduction. But as far as some of these other unclassified major groups go, we're not entirely sure. And that's sort of the problem when we're looking at these systems, is that we're not, because they're so unclassified, it's hard to infer what roles they may have, unless they have other well-known sort of species or group descriptions associated with them. But moving on from the Archaea, this is the bacteria. So as you guys can see, it's much more diverse compared to the archaeal sequences that we got. There's a ton more bacteria. Same sort of situation where we have a lot of unclassified major groups of bacteria in these systems. So it's not just the archaea that are unknown, it's the bacteria as well. But if you look at the different roles, we don't see any ammonia oxidizers. And so when we think about the cooperative interactions that they may have, this is pretty exciting, right? We've seen ammonia oxidizers in the archaea and some of the other nitrogen cycling roles in the bacterial side of these samples. So it's the same setup. We've got our coralloids, our mineral crust, moonmilk, our microbial mats up here. And we can see that, we've got Painibacillus nitrogen fixation present in almost all of these samples up here in the green. And so this is pretty exciting when we're thinking about the different nitrogen cycling roles that we may have from these samples in these environments. And when we look into the community comparisons, this was fairly surprising. So these are more Shannon diversity plots. Jenny explained these pretty well, but they're basically comparing the diversity between sample types. We have our coralloids, microbial mats, mineral crusts, and moonmilk. And we can see that when it comes to archaea and bacteria, even though they look slightly different, there is no statistically significant difference between these sample types, meaning that the diversity is essentially very similar across all the sample types across all 3 caves. And this is fairly interesting. At first, I hypothesized that each sample type would have different community compositions, different levels of diversity, and we would see some significant differences in these sample types, but we don't. And this is pretty interesting. And so some other grad student can pick this up and sort of ask some more questions after this as to why we see so much similarities in the diversity of these different sample types. But moving on, going back to this figure, this is the proposed microbial nitrogen cycle in these caves. So I've taken all of the nitrogen cycling associated groups of bacteria and put them back into this diagram. So if the font is yellow, that means these are archaeal groups. If it's white, that means it's bacterial. So we have nitrogen fixers, we have ammonia oxidizers, we have nitrate reducers, but we could not find any nitrite oxidizers. And this raises a ton of questions. Are they just not there? Unlikely. What I think is going on is that it's hiding in some of those unclassified groups of bacteria and archaea. And we just don't know the potential roles because we don't have enough information in other databases to really ascertain what the nitrogen cycling roles of some of these major groups could be. But another interesting question is, why do we have so many archaeal ammonia oxidizers and no bacterial ammonia oxidizers? And there's a couple hypotheses for this. One, archaea could just be better situated for these extreme environments in terms of oxidizing ammonia compared to their bacterial counterparts. This could also be, unfortunately, a limitation of our methods, meaning that we just did not get sufficient sequences to really show any other bacterial ammonia oxidizers, or they were in low counts. So we didn't really see them when we looked at the top genera. There's a couple explanations, and so more questions for another grad student to kind of go into when it comes to these odd results that we're seeing. But we almost have the full nitrogen cycle. And once we sort of run some more robust analyses of these different sample types and these DNA sequences, maybe we will find more nitrite oxidizers and sort of complete these nitrogen cycles in these systems and get more information out of that. I've got 5 more minutes, so I'm going to kind of go through this. So future work, we're thinking of doing a co-occurrence analysis. And so this would be particularly cool because it'll basically tell us what bacterial species show up with other bacterial species or archaeal species at the same time. So when we think of this nitrogen cycle, if these guys are creating byproducts for one another to use cooperatively, ideally, if you have ammonia oxidizers that are going to produce nitrite or nitrate, they should show up with nitrite oxidizers or nitrate reducers since they're producing what they need for their own metabolic processes. So a co-occurrence network analysis should be able to show us these potential relationships between the archaeal and bacterial species. And it may give us information on the nitrite oxidizers that we're missing in our nitrogen cycle by showing some unclassified groups showing up with other bacterial and archaeal groups that oxidize ammonia and things like that. Additionally, we're going to do a stable isotope analysis of additional nitrogen sources. We're going to look at the atmosphere, leach surface water, and surface plant roots. All of these have unique isotopic profiles. And by analyzing them and then comparing to the isotopic profiles in the actual microbial communities, we can sort of see where they're sourcing their nitrogen and how much of that source they're integrating into their own cellular mass and tissue. So it'll give us a good idea of where they're sourcing their nitrogen and how these values can fluctuate in these systems. And in turn, just tells us a ton of information about these microbial communities and how they continue to shape these environments and contribute to secondary speleothem formation. But are there any questions at all? Yes.
So, initially, you have such diversity, how are those getting into the cave? Through the atmosphere, the water, and the roots?
So there are probably a handful of ways. They could be coming in from critters coming in and out, just shedding, just bacterial contamination in that sense. It could be coming in from leached surface water. It could be coming in from the roots as well. They're not likely, they could be getting blown in soil that just comes into the cave itself. And then there's this very intense selection process that happens afterwards, right? Just because something can survive on the surface does not mean it's going to be able to survive in these cave environments. But once you, depending on where that input is from, after it goes through that selection process, whatever is able to survive in these environments stays and persists and just joins these fairly intricate microbial communities. Yeah.
Well, one question I had, and I know it's maybe a side question, but it's just when you find all these unknown species and stuff, like how is that shared with the rest of the scientific community? Well,
we have to take them and submit them to the database, NCBI database. That way we can make them available for other people to sort of look at, and hopefully as more people conduct more environmental microbiology analyses, we start to see some more hits on those unclassified sequences, and that can tell us more about what their potential roles are, not only just in these cave environments, but maybe in others as well. Because just because we find them here doesn't mean we're not going to find them in other areas as well. Yeah. This cave, well, these caves are approximately 750 years old. Yeah.
Not that old, like
No, not at all. Yeah, they're much more different environments. They have much more connectivity to the surface, and they're far less oligotrophic than you may expect with karst environments that are very deep in the earth. Any other questions? Yeah.
Bacteria identified at all aerobic?
No, they are not. They can be aerobic or anaerobic, and particularly with the microbes that sort of produce minerals on their membranes and sort of conduct these precipitation reactions, they're kind of burying themselves. And you can have this sort of spatial diversity as you look at these different deposits where some are in anaerobic regions, others above them are more aerobic, and they sort of work together to compartmentalize themselves and also provide nutrients that one may need, in this case with the nitrogen cycle, with the different byproducts of the different processes. Any other questions?
And where these caves are, like in that lava flow, we are just up the hill like a mile.
Yep.
And we are on 1907 flood. So that Mauna Loa is constantly erupting. And so there are ones that are in newer and ones that are older.
But yeah, and I'd also like to thank the National Speleological Society, the Cave Research Foundation, Karst Waters Institute, and Cave Conservancy of Hawaii, and of course, UNM Biology for both funding these projects and giving us permission to enter these caves. Yeah. These lava caves are very important to the people of Hawaii. They were used as ritual sites and burial sites, things like that. So we really want to thank the Kupuna and the landowners for allowing us to go into these environments. And we really tried to respect their history and significance as we were moving through them, not to destroy too much of what could be culturally significant. Yeah.